try to make this an interesting lesson. Some of this is a refresher, but I think you're going to get some new stuff today. So let's dig in. Um, what are some wavelength effects on plants? So, I mean, basically, what are the light effects on plants? Obviously, photosynthesis is a key player. Flower initiation, photoperiod, we've talked about that. Initiating germination, we've talked about that. We've alluded to internode length, where we talk about controlling crop heights, but we spent more time focusing on just light intensity and temperatures to control that. Fruit maturation, so we'll dig into that a little bit with tomatoes. Seedling de-etiolation, that might be a new term for some of you. Probably should have been covered in Plant Science 1, but doesn't matter. That's when the plant is triggered to produce chlorophyll. Stomata and chloroplast movement, that's probably new today. Shade avoidance, we've talked about that, but there's maybe a little bit of a twist on it. We've alluded to green light and photosynthesis. I'm going to introduce you to the Emerson effect. I think I mentioned it briefly in one class. We'll look at it again today a little more clearly. And then, of course, disease control. This is looking at just sunlight. The red line kind of gives you an idea of where most of the energy is in the spectrum that comes from the sun, and quite a bit of the energy is in green. So I guess keep that in mind, right? That's a predominant color. In terms of micromoles, this is just measuring units that are useful to plants out of what's all there, but let's look at maybe a better graph. I brought this up in another class where this study was done where progressive wavelengths were exposed to the plants while they were in the presence of radioactive carbon dioxide. And then afterwards, the plant was destroyed and they isolated the radioactive carbon dioxide. And the idea was that any carbon dioxide that was taken up by the plant, in other words, incorporated into sugars through photosynthesis, would have been relative to how useful that light wavelength was at that time. So starting with UV going all the way over to far red, the carbon dioxide levels were assessed. And essentially what it shows is that, yep, there's definitely a peaking amount in the blue area. The middle is still useful, but not quite as much. And then it peaks around red, orange, and then drops abruptly in terms of the wavelengths that were actually driving sugar production. And we mentioned this in that other class that, green is still of use, but a lot of LED lights target blue and red. And I think I mentioned in that class that that's primarily because of energy efficiency. These two LED elements are fairly energy efficient and they're inexpensive to buy, which makes them easy targets for making really simple grow lights. Beyond photosynthesis, what are the pigments that plants use? So chlorophyll, obviously, we've talked about that. Phytochrome, we've talked about that. A new one today would be cryptochromes. We haven't spent a lot of time talking about flavonoids and carotenoids. And then lastly, phototropins, which is probably also new to you. Let's look at these. Chlorophyll lines, there's the blue area and the red area, pretty straightforward. Carotenoids absorbing in the blue, cryptochromes in the blue UV spectrums, just to put things in perspective. And again, I said green is not entirely useless to plants, so that's a spectrum we want to make sure we don't forget. And I think right towards the end of the lesson, I have some pictures from a study that actually looks at green light that's kind of interesting. Yeah, I think I've beat the green color thing down enough. Let's move on. So the Emerson effect, this is a really interesting phenomenon. We've known about it for quite some time. I think it's interesting that a lot of new LED light companies are not aware of the Emerson effect. Maybe that's just an example of them not doing their engineering research. I don't know. But when they shine red light at a plant, again, this is relative fluorescence. This is looking at CO2 uptake. Okay. The relative fluorescence for far red and red, if you added them on top of each other, would be about this amount of total photosynthetic activity. But when you combine far red with red, we actually get an additive effect, which makes photosynthesis more efficient. And this was after it was named after the person who discovered it, Emerson, obviously. And it is something that makes certain grow lights more efficient than others. And of course, what my favorite grower light, HPS lights, produce ample far red light. So we are definitely benefiting from the Emerson effect in those lights. So if it's an LED light, you need to make sure that there is a peak up in the 730, 720 to 740 nanometer range in order to achieve the benefits of the Emerson effect. And of course, the amount of far red produced, makes a difference too. So you don't want just a tiny bit, you need a fair amount. And unfortunately, far red LEDs are not very efficient at producing huge amounts of photons. So that can be a real challenge. Then looking at far red, we have talked about this before, shade avoidance. Just a reminder, because I want to make sure you guys know how this works, right? So the idea is that a plant that has nothing over on top of it will receive the same amounts of red and far red proportionately. Now, when the plant's underneath a leaf, the leaf blocks the red or quite a bit of it, but the far red penetrates, which causes stretch. So that's how the plant senses if it's being blocked, if light's being blocked or shaded on, and then it will stretch to try to get higher than whatever's shading it. That's basically that competition that happens in the forest understory. So shade avoidance is a critical response, and we need far red light for that to happen. So again, if you're looking at grow lights, if you don't have far red, you end up with an exaggerated uneven crop if they're close. So some plants will excel and others will become weaker. Whereas if you have far red, the crop will grow a little more evenly as they all compete with each other to be at the top. So the effect is a nice uniform canopy, which has all sorts of other benefits. So worth keeping an eye on that. And again, green light. Obviously, green plants are green because they reflect green light. And that works in our favor here because any light that happens to bounce down into the understory is in fact useful to the plant. So green is not entirely wasted. The fact that it bounces down and gets lower into the canopy means that lower leaves can still make sugars, which is tremendously useful if the plant's trying to make absolute use of everything that it gets as much as it can. And of course, studies have shown that there is increased biomass with the addition of green in the spectrum. So now, just a couple of little green LED lights may not make a huge difference. It's hard to say. That study at the end has up to 20% or so. So that's a fair amount. Some LED fixtures put just 2 green LED lights in and they think, well, there you go, we got green. But, that's not going to cut it. Looking at blue light inhibits hypocotyl stretch. So remember, when the seedling germinates, the hypocotyl is what's going to push the cotyledons up through the surface initially. And depending on how much light you have or the type of media, blue light is essential to keep it compact. So this is looking at seedling production, where we're trying to keep the crop's consistent size. We need to make sure that we have sufficient blue light in the spectrum so our seedlings don't get stretched. And as far as stem stretch, this is a snippet from another study that has 25, 50, 100% blue. And you can see that as soon as there's 25% blue, we have pretty good height control. So we don't need a ton of blue light, but 25% of the entire spectrum makes a big difference in terms of keeping your seedlings compact. And I think this is where I've said consistently that LED lights are most commonly seen in propagation areas where growers are trying to keep young plants from getting stretched. Case in point, with this pushed off spring, growers are really trying hard to keep their plants to slow them down because they know the market's delayed. So being able to use high blue content LED lights would preclude the use of growth regulators to keep your seedlings short, which might hold off on transplant dates and might keep your crop at a peak by the time you need to sell it, which will obviously be somewhat reduced. This is just a close-up with some more details on that study. These are impatiens plugs that they used, but I'm going to skip on past this. This is a picture showing de-etiolation of seedlings. So not only do we have lots of hypocotyl stretch because of the lack of blue, but because blue also triggers the development of chlorophyll. So they are white. And I think we've seen this before. I believe microgreens that are grown from corn, we typically like to grow them in the dark because then they don't produce chlorophyll, and that makes them taste really sweet. I don't know if you've ever had blonde corn microgreens, but it's like eating liquid sugar. They're pretty amazing. Easy to grow at home. Can make a pretty good business without much light. Look at that. Just growing sweet corn microgreens. One other place you would have seen this would have been, say, if you've ever moved a rock or something. Say you laid down a floor mat on your lawn for a few days and you pick it up. Obviously what happens is with the lack of blue light, the plants actually degrade the chlorophyll and reallocate it elsewhere. And so then when you pull the mat away, the leaves are all yellow. In this case, the grass has gone yellow. Within a couple of days, of course, with regular sunlight, there's plenty of blue, so they will actually redevelop chlorophyll and go green again. There's even a weed here germinated that's all yellow with lack of chlorophyll. Blue light also triggers the production of anthocyanins. So anthocyanins are a purple pigment. We've talked about them in terms of phosphorus deficiency, right? So when you have no phosphorus in your crop, the crop tends to go purple. That's because anthocyanins are a stress response molecule, right? So if the plant's ATP is involved in energy transfer, and if the plant can't move energy properly, there's going to be an excess of electrons. Those electrons cause photooxidative damage. Again, we've talked about that before. Plants cannot handle that. Those electrons will start killing the leaves. You'll get necrotic spots, burns, blotches on your leaves. So the anthocyanin helps to regulate that and minimize the burn on the leaves. There's obviously consumer appeal in that as well, because if you're selling a red leaf lettuce, obviously that's genetically selected to overproduce anthocyanin. There is this idea also that anthocyanins are beneficial nutritionally to people because of their antioxidative properties. That may be true. It's hard to say how long the anthocyanins actually stay in your stomach before they're broken down by your acids and things like that. But you know, it can't hurt. This is a close-up picture of lettuce. So on the left we have grown under white light, under red LEDs, and then under blue. White light is the broad spectrum. You see a lot of LED lights sold as white light. They're nice, but white lights, I think I mentioned, are not very efficient when it comes to LEDs because of how you have to dope them to make all those spectrums. You're better off hand picking the LED elements for the spectrum you need, just making sure you select the correct elements. That's probably the most critical aspect when it comes to LED development. Another interesting side effect of blue, or benefit I suppose, is that in the Solanaceae family, which of course is important to us in the vegetable world, if they don't have blue in the spectrum, especially tomatoes, you get edema on the leaves. I don't know if you remember us talking, we didn't really get into it, I think, because we were kicked out of the college at that time and we didn't have plants, but ivy geraniums are really prone to edema. I mentioned that in an online class where when you have high humidity and the mesophyll cells begin to swell and they actually push through and rupture through the epidermis of the leaf, causing these bumps. In the Solanaceae family, if you do not have blue, you actually get edema as well, not just on the bottom, but on the entire surface of the leaf. Um, a lot of people who have grown tomatoes under LED lights will begin to see edema. And that is, again, because they're missing blue in the spectrum. So I don't think this phenomenon is fully understood. We just know that they need blue light. I always think it's cool coming across stuff that we don't know yet because, although these days it's becoming obvious that we don't know much. um, Phototropins. Now, this is an interesting class. We never really talked about it in Plant Science 1, but in the far red study that looked at phototropisms, where plants move towards light, it's actually phototropin is the protein that's involved in that process. And it's actually not just far red, but it's also blue light that triggers that response. And I believe blue light has an even more pronounced effect on the movement of the plant. But from a really interesting growing perspective, blue light causes stomata to open. Now, I tried hard. I could not find the graph outputs from when in the Canvas program we did a really interesting project with our first group of students. Because it's an indoor facility and it's relatively sealed, we also had lights where we could control the spectrum. And we did an experiment where we hit the plants with far red in the morning for an hour and a half and then went to full spectrum. And then on another day, we hit the plants with blue light for an hour and a half and then went to full spectrum. And we tracked the carbon dioxide levels in the bunker. And what was actually happening was the blue light, true to its description, it was actually causing stomata to open, and we saw CO2 levels, which were fairly high at night, sharply dip in the morning when the blue light was on. Whereas when the far red light came on, there was no change whatsoever until the full spectrum light came on, and then it dropped. And what we noticed was that if we timed it right by having blue light, we could actually take advantage of the full spectrum emission light sooner and get about another hour of productivity out of the day. So I don't think anyone's ever done this before. We didn't publish it. Ran out of time, and I'm not in that stuff anymore. So, but anyways, there you have. Chloroplast movement. This is a weird one. Chloroplasts, I guess you try to see chloroplasts as almost like a bacteria inside a plant cell. So it's basically almost like a cell inside a cell. And they can actually move around. They're not totally rigid. If light levels get too intense, now we've talked about photoprotective molecules like anthocyanin, but blue light is a key indicator that intensity is really increasing on a plant. It will actually physically drag its chloroplast deeper down into the cell and trying to reduce the intensity of light that they're being exposed to. Because of course, if they have too much light hitting them, they can't control the production of electrons, and you get photooxidative damage, which we've mentioned. So the fact that blue light actually causes the movement of chloroplasts is pretty cool as well. It's just a summary slide talking about how phototropism is involved in plant movement towards light, that's blue and red and far red. I introduced you to cryptochromes, right, where we deal with deetiolation, and phytochromes. We know about that because those are involved in sensing short day, long day. There's actually evidence that blue light plays a role in this as well. It's only been done by a couple of studies. That's terrible. I'm learning how to write with my right hand, though. That shows blue light enhances photoperiod detection for crops. So there's some thought that if by including blue in the spectrum for photoperiod, you can actually reduce the electrical requirements for supplementing light to enhance your photoperiod. So maybe as electricity gets more and more expensive, people will start to play with this. And when we have cheaper LED lights where we can play with the spectrum. But I think in your lifetimes, you guys will will see this kind of level of control for growers. And, the more you can tweak it, the more electricity you can save, the more efficient your operation, right? Um, Fruit ripening. This is one that takes red, far red, and blue. What's interesting about this is as fruits ripen, their chlorophyll content decreases. This is a lot like leaves in the fall, right? The chlorophyll is broken down, the nutrients are extracted and reused. Same thing with tomatoes, back into the vine and reallocated. And in the process, carotenoids, they're present the whole time, but they're upregulated. So there's more of the carotenoids present as the crop ripens. And that is enhanced by having more red and more blue. So we want more red and blue to achieve this, which is why it's important to de-leaf above your fruit. Now, I've noticed a couple times this year the leaves have been very aggressively ripped off the plant. I mean, we do have to remember the plants need leaves to ripen. But certainly not having the tomatoes buried in leaves is going to accelerate their ripening, which obviously means you get your crop off quicker, which is good for business. In the wine industry, they regularly de-leaf around the grapes. You'll see towards the end of the season that the leaves are gone and the clusters are hanging separately at the bottom. That's also because as the grapes are ripening, that same red and blue spectrum reduces molecules in the grapes that are undesirable and increases the molecules that are desirable. Things like the pigments, again, they're mostly anthocyanins, but there's a bunch of classes of molecules that, grapes, they're always so complicated, but it doesn't really matter. The point is getting the red and blue accelerates their ripening. So in the tomato industry, we rely on that. In the pepper industry, we rely on that. More so on the tomatoes and the peppers, but definitely makes a difference. Looking at the UV end of the spectrum, plants can't really use it. They use some of it in the cryptochrome spectrum. There's some overlap, but I want to talk about UV from the disease control perspective because it's kind of interesting as we move away from certain chemicals and we look for alternatives. UV has become very interesting. It's not entirely new. It dates back to the 1960s, actually, where tractors would drive with giant lights behind them and shine them under orange trees. But let's look at this. So UV causes DNA damage. In this lesson, we've talked about photoprotective molecules like anthocyanin. These molecules are not present in fungi, which means that when the UV light hits them, the DNA is damaged rapidly. So we're looking at just brief, intense exposure is sufficient to kill surface-dwelling pathogens, right? Now remember, with most pathogens, not all, pathogens, but the ones where spores land on a surface, the hyphae will emerge and grow across and down into the tissues. It's that early stage where we're trying to catch them. Once the fungus has penetrated into the plant's tissues, obviously the plant's own photoprotective molecules will protect the fungus. And then you need to look at systemic approaches to kill the fungal infection. But if you are regularly exposing your crop to UV light, you can actually kill the hyphae as they emerge from spores. Let's take a look at some modern farm examples. This is a UV arc that goes over grapes, and the driver just drives along the rows at night and illuminates the grapes with the UV lamps. And at a slow pace, that is sufficient to cause enough genetic damage to kill any developing powdery mildew spores and Phytophthora. This is a rose grower, and these are robotic booms that go back and forth and down the rows at night to illuminate the roses, which are obviously very susceptible to powdery mildew. Roses have benefited greatly. One of the challenges they have with this crop is that the roses, as you can see in the picture, are very dense. There's a lot of foliage. So you're only going to get control on exposed leaves. But, maybe in the early stages, if you just keep it clean and then you just have to stay on top of it, it's hard to say. Or if they have enough intensity, maybe it's able to bounce around the leaves and get sufficient penetration. This is a robotic UV lamp for strawberries. So these are hydroponic strawberry troughs. and this bot just goes back and forth between the rows. We actually built one of these a couple years ago at the college, and it's in my garage. I got to fix it and bring it back. But yeah, these strawberries are very prone to powdery mildew, and these particular devices are a huge huge benefit to crops like strawberries, where powdery mildew is a big problem. And on top of that, there's botrytis in the fruit, things like that. So it's fairly inexpensive to do, and these days you can even get UV emitting LED lights, which are slightly more efficient, but I don't know how long they last. I think cathode discharge are still the cheapest, which most of these are cathode tubes. Okay, we're near the end. This is just an interesting paper. This link works if you're looking at my other presentation that's actually PowerPoint, not video. But if you click on it, it'll take you to this magazine article where they studied this. But what's interesting is the green and red light produced a decent amount of biomass for green lettuce. But adding the blue gave us more pigmentation. And just leaving the green out altogether gave us the most anthocyanin, but we didn't get quite as much biomass. Although it doesn't look too bad, but this head looks a little more dense. Now, it's just one little study. I don't know how stringent it was, but I thought it would be interesting for you guys to have a look at it. You may want to look up the magazine article. One last thing I wanted to talk about is measuring chlorophyll in plants. This is a simple device. Some farms have it, some don't. It allows you just sandwich a leaf between a light source and the sensor, and the light passes through the leaf and it measures how much is absorbed by chlorophyll. That tells you how much chlorophyll is basically in the leaf, which means if you are doing it on a regular basis, you can see if there's reduction, which tells us that you may have the beginnings of a nitrogen deficiency long before you actually see it with your own eyes, which of course is what you want. You don't want to wait till you have yellowing leaves. You want to keep on top of it. And if you're trying to run a nutrient use efficient program where you're not overdosing nitrogen, this is the tool that's going to let you do that. Something to keep in mind. Anyways, I'm done. Take care.